Hi, my name is Trisha Savam, and today I'll be talking about how the presence of soil impacts the microbiome of plants. So essentially, Kingdom Plantae has a diverse array of organisms, and they have a diverse amount of habitats. They can live in the water, in forests, in the desert, and even on high altitude mountains. These plants are equipped with different physiological structures that ensure their survival in their habitat, such as different types of cells and different types of root structures that help ensure their survival in their habitats, which is often also aided by specific bacteria that ensure they thrive. For plants that exist without soil, such as the common air plant in the genus Tillandsia, there could be a special bacteria that exist on them that help aid their growth and development. My research question for this is, how does the difference between plants that grow in the soil and not in the soil affect the microbes utilized to promote growth and development? So my hypothesis for this is that air plants in the genus Tillandsia will have microbes that actually help increase their fitness in comparison to regular plants that grow within the soil. So some of the methods that we did in undergoing this experiment was first obtaining a sample. So the Tillandsia ailanthus sample was from a common houseplant, and the blue false indigo was from the Monarch Way Station at UAB. Second, we processed these samples. So to store these samples, we had them refrigerated them in Ziploc bags, and we then crushed this refrigerated sample in a solution with soap, water, 70% ethanol, 2% bleach, and PBS buffer. We then took this sample and we placed it on an LB plate and spread it evenly across the agar. And essentially, what this allows them to do is isolate different microbes and allow them to grow on the spread plate. So once this was done, we took a single bacterial colony and diluted it on another plate, which is known as streaking. And so this allows us to isolate specific microbes in a pure culture, and we're allowed to amplify those microbes. This pure culture will essentially be genetically identical to each other, so we could really amplify the DNA that we want. So we isolate this DNA, and then we want to amplify a specific stretch of DNA known as 16SR DNA, and we had to design a primer for it. And then it underwent PCR for 15 minutes at approximately 37 degrees Celsius. So this creates a large volume of the 16SR DNA, which we then put a small portion of it through gel electrophoresis against a DNA ladder to make sure that product was actually produced from PCR. And then we also sent some of it to be sequenced so that we know the DNA sequence and can cross-reference that with databases to figure out the identity of it. So this is the results that we got from the plates. And for the first plant that we did, the Tillandsia ailanthus, we got 2 bacteria samples, and we only got 1 from blue false indigo. And these are our streak plates that were done. We could see that the blue false indigo didn't have much bacterial growth, but we were still able to isolate a colony from it. So those samples were sent to be sequenced, and essentially we were able to pair it with certain identities. So for the first bacterial sample, we identified it as Pseudomonas nakamosi, which is known as a rhizopheric strain of Pseudomonas isolated from a halophyte. That's what it said on there. And essentially, this strain is known for enhancing salt tolerance and essentially can help plants maintain water. We then also got Bacillus paromycetes, which is known for being beneficial to crops and could provide also bioremediation due to pesticide addition that can inhibit plant growth. And as this is a common air plant that is grown within commercial areas, it makes sense that they would spray pesticides on these plants to help prevent bugs from getting into it and destroying it. So maybe this is a new adaptation or new addition to their microbiome to help prevent inhibition to their growth. Additionally, helping the plant maintain water, the Pseudomonas nakamosi is a good bacteria for them to have because they're not getting water from any like root system or the soil, but they're essentially getting it from like the rain and getting it from moisture within the air. As for the blue false indigo, we found Staphylococcus epidermis, and when initially hearing this name, I assumed that I had my culture go under some kind of contamination, but after doing some research, I realized that it is a bacteria that can have beneficial properties on plants, and some of these include hydrolytic enzyme activity, citrophore production, phytohormone production, and volatile synthesis for inhibiting phytopathogenic fungi. So after all of this, we were able to find some limitations within our study. And like I said, I thought some of the bacteria could be due to cross-contamination, but it also could be cross-contamination from the plants with each other. Additionally, I don't believe enough bacteria was collected for me to really see a big difference between plants that grow in the soil and plants that don't grow in soil. And I don't think there's been enough data collected to come to an appropriate conclusion about how much beneficial bacteria can be on Tillandsia ionantha. And if this is really different from plants that grow in the soil, because they could also have similar bacteria that I found, but it just wasn't within that one leaf that I had collected. And so I believe further research needs to be done. But the conclusion I got from the research that I had done is that it did have beneficial bacteria, and this culture of bacteria can be used on plants that are in water scarce areas and also used for commercial sale, because not only does it help with water like keeping water within, but it also helps with salt tolerance, as well as being able to make sure that plants keep growing in the presence of pesticides. Thank you.